I'm sure everybody will know Elisabeth. She's got this rather scary, otherworldly ability to find errors in images in microbial research. And she's just going to walk us through sort of past exploits and the kind of roller coaster of trying to do this in correct bad science. So she's very much at the coalface. So Elisabeth, I'll hand over to you. Okay, you got everything ready?
Okay, thank you. Can you see my screen? Can you see my slides?
Yeah, everything's good.
All right, great. All right, so I'll go a little bit over first what is good and what is bad science, because I think the distinction between good and bad science is actually not always very black and white. There's, of course, most scientists, I believe, are very honest and hardworking, and I've been focusing on the very bad science. But I think it's also a slippery slope between when science sort of is slipping into the bad. I think most of us are biased. We all have our ideas of how our results should look like. We all think about the outcome of our experiments, and I think most of us, including myself, are tempted to leave out the results we don't want. And we also have experience that scientific studies are often not reproducible. And that is because it's often very hard to capture all the different experimental conditions that we know are important in our studies. But it doesn't necessarily mean that this is bad science. That's still on the honest side of science. But then when we think about errors in statistics and models like p-hacking, then it becomes a little bit worse. And then I'll talk about the bad science, which is even the ugly, predatory journals, fake authors, peer review rings, and paper mills, and of course science misconduct, which is defined as plagiarism, falsification, and fabrication. And as we know, science builds upon science. Every scientist will go and look at the literature. And so if peer-reviewed papers are containing fake news, so like falsified or plagiarized or fabricated results, that means that other scientists will waste their time. And so that is the central cost of bad science, in my opinion, is that fake papers and fabricated papers will waste other researchers' time because they believe that those published results might be true, but they cannot replicate it because it was fabricated. And so I think that is a central theme of why I'm so motivated in finding fabricated and falsified results in published science. I just try to clean up science so that other people know which papers are not good. And what I'm mainly focusing on is biological images, so images in biomedical literature. And most things in nature, including papers in biomedical literature on tissues and protein gels, most of these images are unique. If you look at each of these images, then you can see that all the panels, although maybe similar to each other at first glance, each of these are unique. Even as you can see on the top right, protein bands, so this is a Western blot, all these bands, for me at least, look unique. They all have different shapes and smudges and things that just stand out to me as unique. And so I'm focusing on finding duplicates in these images that are maybe the result of an honest error or maybe the result of an intention to mislead. And so here's some of the examples of papers, published papers with images that are duplicated. You can see on the top left a flow cytometry image that seems to be heavily manipulated. All the areas that I marked in colored boxes are identical to each other, but in between the pink and the orange box, you'll see an area that is different between these two panels, and so that appears to be manipulated. And then on the top right, you see 4 images that at first glance look unique to each other, but they actually show overlapping images. And so at least 3 of the 4 panels appear to have been derived from the same original sample. They just shifted the microscope a little bit to change the view, but I can still find the overlaps. And then there's Western blots that could be shifted, as you can see on the bottom right. And on the bottom left, you'll see some what I call simple duplications where the same image has been used twice. And I've marked these in blue and red boxes. So there's all different types of inappropriate image duplications. And I like to point out that when an image is used twice to represent the same experiment, I call that appropriate image duplication. So I won't raise those. I'm really talking about images that are duplicated to represent different experiments. And you can see that some of these might be honest errors, like the bottom left one where there's a direct duplication. But then the ones that show a shifted point of view or even a manipulated image are very likely to have been done intentionally. And so I did a large study on that. I actually scanned 20,000 papers by myself, not actually reading them, just looking at the images. And I included 40 different journals from 14 publishers. And I found about 4% of these papers to contain inappropriately duplicated figures. And so you could say that's maybe a pretty high percentage, 4%. On the other hand, 96% of the papers did not have those duplications. And yeah, and so you could argue, like, what is the real percentage of the intentionally duplicated or manipulated data? That I don't really know, because half of these, maybe half of these 4% might be honest errors. But even if 2% is intentionally duplicated, has intentionally duplicated results, that's obviously much too high. And actually the real percentage of fabricated papers might be even higher, because I only focus on very obvious things in photos, but there might be line plots, there might be sequence data, there might be all kinds of data that is much harder to look at. You really need to sit next to the researchers in order to catch somebody doing misconduct. So I just look at the very obvious problems in papers, and I'll probably only catch the tip of the iceberg. So just as an example, this is a image that has a lot of duplications. I won't leave it up too long, but if you just let your eyes scan over this at first glance, maybe all these images look different to each other. But in the next slide, I've shown you a couple of the duplications in here. This I cannot imagine that this is an honest error, but yeah, so this is sort of what I do, and this seems so obvious that you could wonder if how could this ever pass peer review? And yeah, I'm wondering the same thing, but it's pretty nasty how it sometimes can be. And so another problem that I've been working with in published science are predatory publishers. So these are publishers that look like they're real scientific publishers. They're usually not indexed in the main indices like PubMed. And they seem to care more about publishing papers for money in the open access model than they care about the quality of papers. So they'll spam researchers asking them to publish in their journal. They offer a very fast peer review and open access model, and they sometimes publish huge amounts of papers on many topics. But their quality, the quality of their papers is really, really low. And they make misuse of the fact that some inexperienced researchers think that these are really great papers to publish in. But they're often misused by lone authors who have very wild or fantastical ideas, and sometimes shady industry companies who will use these platforms to publish papers that look like they're real scientific peer-reviewed papers. And it's really hard for the general audience to distinguish between a real, between quotes, scientific journal and one of these predatory publishers, because some of these look really, really authentic, and the peer review process is really ridiculous. You can see an example here on the bottom, where sometimes these papers are published within 24 hours or even shorter, and so peer review is pretty much non-existent. And yeah, a lot of these papers just are not very good, but they can be misused by a lot of people. And then there's people who publish in real peer-reviewed journals who sort of create their own identity that is completely fake. They claim to work at fake institutions or have fake authors, or they even peer review their own studies. I mean, there's a lot of wild things out there, and if you're as captivated as much as I am, I would encourage you to follow Retraction Watch, because there are a lot of these very strange schemes out there in how people try to game the system. And one thing I've been working on very recently are paper mills. Now, paper mills are sort of, I'm not even sure what they are. Are they companies? There are some entities that cater to the need to publish. In some countries, there's monetary incentives to publish a paper. For example, in China, in your medical school, if you want to finish your medical school and you want to make a promotion as a young doctor, and actually earn enough money to support your family, you're required to publish a scientific paper. And it doesn't really matter if, as a doctor, you're interested in publishing a scientific paper. You just have to do that in order to go to the next level in your career. And so these people are not offered any research time at all. They might not even work in a hospital that has a research lab, but yet they have to publish a paper. And so they buy papers that are fabricated, completely fabricated by what we think are paper mills. So they're just companies that crank out fake papers. And they even use fake figures like the one we worked on the right. And this is a team effort in which many people, I was one of them, but there's many people behind the scenes, some anonymous and some not, who have identified at least 400 papers that all use the same background in Western blots. As you can see on the right, I've enhanced this figure. The bands are, we believe, all fake. They're computer generated, and the backgrounds are all the same. They have all the same stripes and irregularities, and this is how we recognize that. But it just makes you wonder how many fake images are out there that we did not recognize because the paper mills were a little bit smarter and did not use all the same backgrounds. And I've been using very recently on another paper mill that had 121 papers that are identified that all use the same photos and that all all seem to have been fabricated. And so this is obviously a very worrying development, and I've been trying to report all these papers to the institutes, to the editor-in-chiefs of the journals and the research integrity officers. And what has been very frustrating for me, from my point of view, is that a lot of the institutes and the journals do not follow up on these things. And so as sort of a last slide that I want to leave up with some thoughts about these things is like, what are the costs for bad science? So like I said, is that science builds upon science, and every paper that is a fake paper is going to waste other people's time, and it's sort of a brick that is rotten, and that sort of destroys the whole wall of science that this brick is built on. And so we've seen that with the latest COVID-19 papers as well, where we have a lot of new research that's being put out and then immediately retracted. It's very damaging for science, but it's also very damaging for the general audience, who now might think that science is actually not as great as they have always perceived. And so I think as scientists, we really need to think about what that tells the general audience on whether or not science is trustful. And so, I mean, I can talk hours about this, but one thing I would like to point out, it's much better to catch misconduct before papers get published. That saves everybody a lot of time and effort, and we've actually shown that in one of the research studies that we did, that it will save editors a lot of time if they can catch these things before peer review and before publications. How we do that, that's hopefully we can talk about that in the discussion. And finally, I like to mention a lot of conflicts of interest that I've been frustrated about, where publishers and institutes might have all kinds of reasons to not address the problems that I've been finding in these papers. And I would really like to talk to them in order to break through these conflicts of interest and actually correct science. So with that, thank you.
Wonderful, thank you, Elisabeth. Very interesting talk. We have a flood of questions, so we'll do our best to get through them. So first one is for Dr. Bik. Could a program be developed for journals to reverse image search figures in the same way that they use anti-plagiarism software, or do we need to clone you?
I've had that a lot. So several publishers are working on with I believe 3 different software developers that have tools like that. Computationally, it's much harder than scanning papers for text like plagiarism checkers do. So it's computationally pretty intensive, and it's not as good, I think, as the human eye as of now, but it's getting better. So I'm confident that, let's say 5 years from now, or maybe even sooner, we'll have publishers scanning for these things. I'm sure that the bad scientists will find other ways to circumvent this, but yes, that's the best we can do.
Yeah, so you've sort of touched on a question I was about to follow up with, which is sort of 2 questions from separate people, Willem and Joe, and I'll just merge them. So one is, will image manipulators not just get smarter and become untraceable? And the second question is sort of, have you noticed any trends or techniques that are being sort of used to make it more difficult to detect?
Yeah, so to answer the second question first, so as we saw in that one of the paper mills, the tadpole paper mills, we believe that these images were artificially generated. So they have some database of maybe thousands of different Western blot bands, and they generate things that look like gel panels or blot panels out of a database of all these different bands against the background. And because they used all the same background, that's how several people caught these duplications. But yeah, they'll if they had done something different, they might have not been caught, and that's what I'm worrying about. That we as data detectives are only catching the tip of the iceberg, and I'm very worried about how we can ever catch these things. And one of the obvious things that we should work on is trying to work with different models of how we judge people's career and resume is maybe by not looking at the output, not looking at papers. But yeah, that's something that we have discussed before in this panel. But it's going to be a very tough call to find these things as computer techniques become easier. It becomes easier to fake things. So it's going to be very tough to catch this.
James has asked a question that I wanted somebody to ask because this is sort of not fitting with the usual narrative about journal impact factors. So it goes, in your 2016 paper, lower impact journals tended to have a higher proportion of inappropriate image duplication. Is this finding true more broadly in your experience, and does this provide some argument in favour of journal impact factors?
Yeah, so we did find an association, but it wasn't completely true. We found low impact journals that had very low amounts of duplicated images, and vice versa. I found some of these image duplications in Science and Nature, and obviously high impact journals have usually a more experienced authorship. So the authors who send in these papers they're either much smarter in not getting caught, or they're more honest. I don't know that, but the relationship between impact factor and percentage of these images is not completely, it's not a very linear correlation. It's a little bit, but there's very good journals that have a low impact factor, and there's several that have a high impact factor, and in some of these cases, the high impact factor is actually generated by requiring authors to include papers from that journal to raise their impact factor. So it's one of the peer review scams that we've we've seen.
And I suppose this is a hot topic now because peer review is supposed to be the kind of quality control check, gold standard. But we have a question that's kind of related to that, which is, who are gatekeeping the manipulated data? Is it journals, universities? I mean, at what point can these issues be caught, and who is responsible, if not the researcher, for preventing this from happening?
Yeah, obviously the responsibility is with the researcher, but if you want to game the system, then you're going to do that. And even putting the responsibility at the institute, it's not going to help if the institute is part of the plot, because there's been institutes that really want to raise their university rankings, and they actually encourage their researchers to publish whatever they can in order to get a better university ranking. So if the university is into the scheme, that's not going to help either. So it seems that the most obvious gatekeepers should be the publishers and the journals, but like they do with plagiarism, but they should also be a little bit more open for all the peer review scams and the image fabrication scams that I'm trying to focus attention on, because I think a lot of them might be gullible and they just have no idea that this is going on. And so I hope to raise a little bit the word about that these things are happening and they should be on the lookout for these manipulations.